viral replication. So different stages. First, attachment. The virus attaches to specific host cell receptors, right? Then it penetrates the cell. After penetration, it uncoats, right? It releases nucleic acid. And then you have the macromolecular synthesis. You have the early mRNA and protein synthesis. You have replication of the genome. And then you have the late mRNA and protein synthesis, followed by some post-translational modification of the proteins. And then you have the assembly of the new virus particles. And then you have the release via lysis of the cell or exocytosis. So please know the different stages in viral replication. Bacteriophage. I have a question. What's the function of a bacteriophage? Anyone? Why do we have a bacteriophage?
Integrate into the host cell DNA.
Okay, for example, for gene therapy, if something has cystic fibrosis, for example, you want to modify the gene, right? So you have a bacteriophage to insert the genome into that cell. That's the main thing about bacteriophages. So there are two possible existing bacteriophages. So you have lytic or lysogenic phase. What's the difference? Lytic phase, you kill the host following infection. The lysogenic phase, you do not immediately kill the host. What happens there is you have the integration into the host chromosome, or that cell exists as an independent entity, but you have the virus replicating inside that cell now. So you don't kill the cell at an instant. Contrast this one with lytic, where you kill the host following the infection. So virulent, lytic, lysogenic, temperate. In terms of viral genome, you need to know the Baltimore classification. That's high yield, but on the step, so what to highlight is this: positive nucleic acid and negative nucleic acid. Question: which one is the most dangerous? Is it the positive or the negative, and why? Which do you think is much more infective, much more dangerous, the positive or the negative strand of either
Positive.
DNA or RNA? Positive, specifically your positive single-stranded RNA. Why?
It can be directly used to make mRNA.
Exactly. It can be directly used so that you can have now proteins, right? Contrast this one with negative sense when you need to do reverse transcriptase. Very good. So high yield, positive sense, infective, most infective of them all, most deadly. Now you have double-stranded RNA, you have single-stranded RNA. How does it replicate now? So you need RNA-dependent DNA polymerase or reverse transcriptase. Please know that name. They will not gonna put reverse transcriptase, they're gonna put RNA-dependent DNA polymerase. Reverse transcriptase is commonly used by what virus in the Baltimore classification? Class 1 or 6? Class 1 to 6, which one?
6 and 7.
Exactly, 6 and 7. So your retroviruses, right? Excellent. So know the name. RNA-dependent DNA polymerase equals reverse transcriptase. Also, don't forget that the viral mRNA can also be modified. So you have the 5' to 5' N7 cap, and then you have the poly A tail. Okay, you know viral proteases, they're essential targets in your antiviral strategies. Do I need to know boceprevir and saquinavir right now? No, but term 4, you need to know them. So, okay, this is high yield. Defective viruses. So defective viruses, they cannot replicate without a helper virus. Example, hepatitis B. So it needs hepatitis B virus in order to replicate. Why? Because hepatitis D doesn't have a surface antigen to infect a cell. So it needs the hepatitis B virus surface antigen for entry into the host cell. So high yield defective virus, hepatitis D virus. What does it need to infect cell? It needs a surface antigen of what? Hepatitis B virus for entry into the host cell. Prions. Prions are infectious proteins. They have no genome, RNA, or DNA. So what happens here is you have a it induces the transformation of your proteins from alpha helix to what? Beta, right? Beta pleated sheets. And you know, your beta plated sheets, if they accumulate, they're going to form this vacuolization within the neurons, right? So what are the signs and symptoms that someone who has prions? This is very, very deadly, okay? You have loss of motor control, dementia, paralysis, and encephalitis. You can be dead within less than a year if you've got this prions, your Creutzfeldt-Jakob disease, or your bovine encephalopathies, your mad cow disease. So prions, right? It transforms your proteins from what? From the alpha pleated sheet to beta pleated sheet. If you have an increase in beta pleated sheet, what happens in your neurons? It forms vacuolization within the neurons, thus causing the signs and symptoms that you can see. You have the loss of motor control, you can have dementia, you can have paralysis, and you can have encephalitis and widespread neuronal loss. Viroids, these are plant pathogens, but we're not botanists. Remember, the viroids are the smallest infectious pathogens. They distributed from cell to cell through what? Plasmodesmata. Don't forget that. Viroids, smallest infectious pathogen. And how are they going to be distributed from cell to cell? Plasmodesmata, right? How are we going to regulate your viroids? Sirna. But mainly this is for botany, okay? Right, classification of viruses. We need to classify them according to their morphology, virion size, capsid, yada, yada, yada. You need to know the Baltimore classification. So let's go here. So these are examples of your DNA viruses. So DNA viruses, they have single-stranded, mixed-stranded, or double-stranded. Do I need to know an example of each of the DNA viruses? Yes, that's high yield. You need to know examples. So parvoviridae, human parvovirus B19. This is your slap cheek disease, your erythema infectiosum. Hepadnaviridae, hepatitis B virus. Examples of polyoma, JC virus. Remember JC virus from last term? It causes PML in AIDS patients. Papillomaviridae, you have HPV. Adenoviridae, adenovirus. Most common cause of common cold in children. Herpes viridae, herpes simplex virus, and you have pox viridae, you have your pox virus. So this is another template, DNA virus, envelope, and non-envelope. Question, which one of these survives in the environment? The envelope or the non-envelope one?
None.
Non-envelope. Therefore, which is much deadly? In terms of the lethal dose that needs to be ingested by the cell, which one? Envelope or non-envelope? In terms of lethality, if you say that non-envelope exists in the environment freely, they cannot be destroyed via simple things like desiccation and stuff, then which is much more deadly when it infects the cell?
None.
The non-envelope one. Why? Remember, the envelope viruses, they can be engulfed by your antigen-presenting cells, by your macrophages, right? Contrast this one with the non-envelope ones, when they can just easily inject. They don't have the envelope. They don't have antigenic properties. They cannot be recognized by your antigen-presenting cells, your macrophages, okay? That's high yield. And you need to know the examples, please. You need to know the examples. Properties of DNA viruses, you can read them here, right? Pox virus is the biggest of them all, of the DNA viruses, right? It replicates in the cytoplasm. It's so big, it cannot replicate in the nucleus. Most of them replicates in the nucleus. Now, RNA. Just like a DNA, you need to know examples of each, right? Single-stranded and double-stranded. There's only one double-stranded RNA. What is that? Rotavirus. Rotavirus. Most common cause of diarrhea in kids, children. Rotavirus. The rest, single-stranded. Now you need to know which one is positive and which one is negative. Okay, here's another one, RNA viruses. Another schematic. Properties. This will be emphasized more, I think, in term 5. Term 4 and term 5. Next term and term 5. We're going to go over the differences. But for now, highlight this one. The Baltimore classification, this is high yield. Group 1 to group 7, right? You need to know the properties of each group. You need to know an example of a virus from each group. You need to know the mode of replication for each group. Actually, on the step, they don't use a Baltimore classification, but that's not the point. You need to know this one for your SGU exam. So let's go. Group 1, double-stranded DNA. If you have double-stranded DNA, it goes directly to mRNA and then to proteins. Group 2, DNA, positive strand, right? Group 3, RNA, double-stranded RNA. Group 4, positive single-stranded RNA. 5, negative single-stranded RNA. Group 6, single-stranded RNA. You need to have a reverse transcriptase. And group 7, these are your partial double-stranded DNA. Okay, they also need reverse transcriptase. So we're going to go over each Baltimore classification next. So group 1, as I've said, you have a double-stranded DNA. You need DNA-dependent DNA polymerase to make copies of the strands. And then once you have the positive viral mRNA, it can be converted into your proteins. Example of group 1 viral genome replication, herpes virus, right? Remember, they're non-envelope or they contain a double-stranded DNA genome. Herpes virus, except pox viruses, they replicate in the nucleus. They have a complex life cycle. And maybe I don't know if, did you talk about the different kinds of herpes virus like HHV 1, 2, 3, 4, 5, 6, 7, 8, or not yet?
No, not yet.
Okay, so for now, okay, that will be next term or term 5. So for now, group 1 viruses, these are your herpes viruses. They replicate where? Nucleus, except for one. Which one? Pox virus. Why does it replicate in the cytoplasm? It's too big. It can't fit into the nucleus. The Daltons for that one, it's so large that it cannot be contained in the nucleus. Okay, so group 1, herpes virus. Group 2, you have a single-stranded positive DNA viral genome. What do you need for this to be replicated? DNA-dependent DNA polymerase. It's also high yield to make double-stranded DNA intermediates, and then you need DNA-dependent RNA polymerase to make positive viral mRNA, then which can be translated into your viral proteins. Example of group 2 viral genome replication, high yield parvovirus. Okay, parvovirus. Where do they replicate? They replicate in the nucleus. Don't forget the single-stranded DNA is converted to double-stranded DNA. And then the single-stranded DNA, they produce virions containing either a positive sense or a negative sense DNA genome. Group 2 virus replicate in the nucleus. Example, parvovirus. What do they need to be converted to double-stranded DNA? If it's single-stranded, that you need a DNA-dependent DNA polymerase. Next, groups 3, 4, and 5. Groups class 3, they have double-stranded RNA. They can either be positive or negative. Class 4, positive single-stranded RNA. And class 5, negative single-stranded RNA. High yield here is this one, the positive sense, because you can use this one, single-stranded RNA positive sense. It can be used directly as an mRNA. For the negative sense, single-stranded RNA virus, you need to have transcription. And double-stranded RNA, you also need transcription to make the mRNA. High yield point, single-stranded RNA positive virus, the class 4, it can use directly as mRNA. You don't need transcription for class 4 viruses. What do they utilize? Right? They contain a genome and they utilize RNA-dependent RNA polymerase. Don't forget group 3 and 5, they carry a viral RNA, dependent RNA polymerase. Highlight that one. That's high yield. Groups 3 and 5, they carry RNA-dependent RNA polymerase. And group 4, as I've said, they contain positive sense that can be used directly as mRNA. Now we're going to group 3. Group 3, you have a double-stranded RNA. What do they need to replicate? They need RNA-dependent RNA polymerase to make a positive viral mRNA, which can be translated directly to proteins. Example, I yield rotavirus. Group 3, rotavirus. Do I need to know the mechanism? Not yet, but term 4, term 5, you need to know the mechanism, right? But for now, know the examples, okay? Group 4. Group 4, as I've said, single-stranded positive RNA. It can be translated directly by the host ribosomes to viral proteins. Example of this, high-yield polio virus. So polio virus for group 4, viral genome replication. This is the most common cause of, what do you call this one, for polio? Before, polio is rampant, but because of the vaccine, it has decreased its sensitivity in the United States for this one, for the polio virus. But in the developing world, it's pretty rampant. Group 5, single-stranded negative RNA. So what do they need in order to make proteins? RNA-dependent RNA polymerase. You need to know that one to make viral proteins. Example of group 5, rabies virus. These are your rhabdovirus. Term 4, you're going to learn about paramyxovirus. Example, your respiratory syncytial virus. You can also have segmented virus, but these are low yield. Bunyavirus, arenavirus, orthomyxo virus. So they must carry what? For group 5, they must carry their own RNA-dependent RNA polymerase, high yield, okay? They need to carry RNA-dependent RNA polymerase. Example, rabies. Group 6, so you have a positive single-stranded RNA viral genome. What do you need in order for this to be converted to double-stranded DNA? I yield RNA-dependent DNA polymerase. Your reverse transcriptase now to make double-stranded DNA intermediate. After you've made this one, a double-stranded DNA, what happens? What do you need? You need, again, DNA-dependent RNA polymerase. See the difference, right? To make positive sense viral mRNA that can directly be translated into viral protein. As you can see here, right, what's the schema of the classification? If we have a positive viral mRNA, this can be translated directly into the proteins. Group 6, you need RNA-dependent DNA polymerase to convert it to double-stranded DNA. Example, high yield is your retroviruses HIV. Question, did you talk about the attachment of HIV? How does it attach and how does it affect the machinery of the host cell that it infects? Yes or no? Anyone? Did you talk about the exact mechanism of the HIV replication, the life cycle of the HIV virus?
I don't think so.
Oh,
It was a fusion or endosome acidification or something.
no, okay. So I don't want to get into like details for this one. Did you talk about GP120, GP41, P27, or not yet?
No.
Okay, so the main point here is this one, right? First, you have fusion, right? It fuses with the host cell, and then you have the virus has reverse transcriptase. The reverse transcriptase, it catalyzes the synthesis of a DNA strand complementary to the viral DNA, and this one, the hybrid RNA-DNA hybrid, goes to your nucleus now. So this double-stranded DNA it is incorporated as a provirus into the host cell. And then these are transcribed to RNA molecules, and then it can infect other cells via exocytosis. I think that's for next term or term 5 for the HIV replication cycle. But the main point here is this. What do they need, right, in order to replicate? RNA-dependent DNA polymerase. This is high yield or reverse transcriptase. Group 7 is a partial double-stranded DNA, so HDV, right? In partial double-stranded DNA, and then you have in the nucleus, you have completion of the double-stranded DNA. Example is Hep B virus. Hep B virus, they are enveloped double-stranded DNA. They contain partial single-stranded DNA. And what do they need in order to replicate? RNA-dependent DNA polymerase. These are your reverse transcriptase. Okay. Another example, class 5. This is your negative single-stranded RNA of Hep D. It just shows you that this is a defective virus and it needs the surface antigen of your hepatitis B in order to infect a cell. That's it. Next term, you're going to learn about the life cycle of each of these hepatitis viruses. So there are different kinds of interactions between viruses, right? First, recombination, high yield. For recombination, there are two viruses, right? And then they recombine. If you put HSV1, for example, and HSV2 together, there's a chance that they recombine when they infect the same host, okay? So recombination, you have an exchange of genetic material from HSV1 and HSV2. You have this recombination and you have new strains now of HSV. We call that one recombination. Contrast that one with complementation, right? So you have two viruses. They simultaneously infect the host cell, but at different gene location. So they can successfully propagate. Why? Because they complement each other and they rescue each other's defective functions. Think about complementation, HBV, and the defective virus is HD, right? H delta compared to H beta, right? So they complement each other. The surface antigen of HBV is given to HDV in order to infect a cell. That's why you usually have co-infection now. Next term, we're going to learn about the difference between superinfection and the co-infection. Co-infection, you have the same time. Superinfection is you have first HBV, and after you're cured with HBV, you're going to get HDV. So there's a difference, but you're going to learn that next term. So that's complementation. They complement each other. They rescue each other defective functions. Okay, high yield antigenic drift versus antigenic shift. What's the difference? Drift, right? You have a small changes in the genome, right? Within a definite strain, it occurs at what? At slower rate compared to your antigenic shift. For example, influenza virus and rhinovirus. That's why we get flu vaccine every year because of this antigenic drift. They are just small changes in the genome, right, which occurs at a slower rate. Now, antigenic shift, this results in what? Reassortment of RNA segments, right? So this is, which one do you think can cause pandemics? Is it the antigenic drift or the antigenic shift? Pandemic, right? Like the coronavirus pandemic. Which one? Is it the drift or the shift?
Shift.
Shift, exactly. Epidemics. Epidemics. Which one can cause an epidemic?
Drift.
Drift. Excellent. So antigenic shift is the main cause of your pandemic. Remember why? Because it results in reassortment of your RNA segments between two different viruses, right? However, drift, there's only small changes in What is this? Wait, I'm sorry. Skype notifications. Never mind. So it occurs at a slower rate. Okay. Next, phenotypic mixing. Just know the terms. Know that phenotypic mixing or transcapsidation, the genome of the virus is randomly incorporated in the capsid composed of proteins from different or both viruses, right? For pseudotypes, they are formed when the nucleocapsid of one virus acquires the envelope from another type of virus. So the questions were going to be definitions, okay? Know the definitions of the two for the phenotypic mixing. Okay, next, let's talk about protozoa. So that's the viruses. Let's summarize. You have the different, you know, the Baltimore classification from group 1 to group 7. Know their characteristics. Are they single-stranded DNA? Are they double-stranded DNA? Know examples of each of the classification. Which one is deadliest? Which one is the most infective? The positive sense, group 4. Positive sense, single-stranded RNA. Now, let's go to protozoa. And I remember Amoeba Sisters. They had a great YouTube channel for learning biochemistry and cell biology from YouTube. Okay, protozoa, what are they? They're unicellular. They're animal-like microbes. They most reproduce asexually, right, by a binary fission, or they actively feed on the cell via a trophozoite, right? A few others, like your Plasmodium, which causes malaria, they have a complex life cycle. They have asexual and sexual phases, but they are unicellular and they are animal-like microbes. Some of them has motility. Okay, some of them, you can get them via fecal-oral route. The infective forms, if they're fecal-oral, your cyst, trophozoite, and your oocysts. If they're urogenital, then they are acquired via sexual transmission. So the infective form is going to be trophozoite. For example, your Trichomonas vaginalis, right? You have a greenish discharge in the vagina. You have a strawberry cervix. That's a Trichomonas vaginalis. For blood and tissue, of course, it's by insects. So the infective forms are going to be trophozoite, oocyst, promastigote, or trypomastigote. Do I need to know which one is intestinal, and do I need to know which one is extra-intestinal? Well, it helps, right? So for intestinal, think about amoebiasis or cryptosporidosis, giardia, microsporidosis. For extra-intestinal, your trypanosomas, your chagas, your leishmania, your kala-azar, your malaria, your toxoplasmosis. But we're going to go over the most important ones. So put a star here. How do they replicate? Put a star here, intestinal protozoa. What's the generalized life cycle of each? You're going to ingest them since they're intestinal via what? Cyst or oocyst, or directly as a trophozoite, right? If you're going to ingest them as a cyst or an oocyst, you have encystment into the body, which forms a trophozoite, and then encystment, then going back to cyst. They can be passed into feces. They are resistant to hostile environment, the cyst form. They are resistant to hostile environment, and they do not multiply. If you ingest them directly as a trophozoite, they are metabolically active. They are motile, and they multiply by replication. Intestinal protozoa, you can ingest them via a cyst or an oocyst, or directly as a trophozoite. If it's a cyst, it's passed into the feces, non-motile. It is resistant to hostile environment. It does not multiply. If you ingest them as a trophozoite, they are metabolically active. They're motile and they multiply by replication. Examples. Okay, so for this one, you need to know the images. They're going to put some images there and you need to identify. Remember, the images are there to help you. They're going to give you signs and symptoms in the vignette, right? And you already know your diagnosis, but the pictures are there to help you narrow down your diagnosis. Let's go. One by one. Entamoeba histolytica. What does it use for locomotion? High yield pseudopodia. Underline that one. It uses pseudopodia for locomotion. It invades the colon. What does it cause? High-yield bloody diarrhea. This is your amoebic dysentery. It also causes amoebic liver abscess, okay? And it reproduces asexually. Next, Balantidium coli. Look at the image here. You have cilia, right? Underline that one. It's a motile ciliated parasite that lives in the colon of the pigs. How does it reproduce? Asexually. Giardia lamblia. Look at the picture here on the right side. It uses what? It flagellates. It has flagella, right? It's a flagellated protozoan. It lives in the small intestine and results in malabsorption. Malabsorption. Reproduces asexually. Trichomonas. Look at the right side here. The image of your trichomonas. They have flagella. They're a flagellated urogenital parasite. They're sexually transmitted and they reproduce asexually. Cryptosporidium. Are they motile? No, they are non-motile. Immunocompromised, more prevalent in immunocompromised patient. The most common cause of diarrhea in someone who has HIV, AIDS is Cryptosporidium parvum. Cyclospora kyatinensis. Are they motile? No, they are non-motile. They may cause diarrhea for several weeks and they reproduce sexually and asexually. So for each, know the image. What do they use for locomotion? The general characteristics, right? The disease that they're causing. And if they reproduce sexually or asexually. Let's summarize again one by one so that we can have a clearer image, right? What's important? Entamoeba histolytica, pseudopodia, bloody diarrhea, asexually. Balantidium coli, cilia, asexually. Giardia lamblia, flagella, asexual production. Trichomonas vaginalis, flagella, sexually transmitted, asexually. Cryptosporidium parvum, non-motile, right? More common in immunocompromised patients. Both life cycle, sexual and asexual. Cyclospora kyatinensis, non-motile. Diarrhea, they're both asexual and sexual. So you might ask me, how can I differentiate Cryptosporidium parvum from Cyclospora kyatinensis? Well, if in the vignette it says immunocompromised, then it is Cryptosporidium parvum. And look at the image. They're different in their images. Okay? Next, another pretty picture of your trophozoites and your cysts. Trust me, they're going to put some images there. So this is just look for this in your spare time and memorize the images. Now let's go to extraintestinal, your blood and tissue protozoa, your plasmodium, different kinds, Falciparum, Vivax, Ovale, and Malariae. I have a question. Did you go over the differences between each types of plasmodium or not yet?
No, I don't think so.
Not yet.
Not yet. Okay. For now, remember that your Vivax and Ovale, they have hypnozoites in the liver, right? So they need something else than chloroquine, okay? They need something else than chloroquine. And Plasmodium falciparum is the deadliest of them all. It can cause hemorrhagic fever for this one. Plasmodium falciparum, the deadliest of them all. P. malariae, well, tertian fever. And Vivax and Ovale, they have hypnozoites. Hypnozoites, is that how you pronounce it? Sorry. So that's how you'll think about that for the Plasmodium. And they are transmitted by what? Anopheles mosquito, specifically the female Anopheles mosquito. So early in the morning and later in the afternoon, don't go out, right? Especially summer, because the female Anopheles mosquito, that's their feeding time. Okay. Next, leishmania. Leishmania is transmitted by sandflies. It can lead to different things. But the high yield here is the cutaneous leishmaniasis and the visceral leishmaniasis. Do you talk about Kala-Azar or no?
No.
No. Okay, never mind. That's going to be on term 5, I think, or term 4. So leishmania transmitted by sandflies. It can cause visceral and cutaneous leishmaniasis. How about trypanosoma? Trypanosoma in Africa, sleeping sickness. Sleeping sickness transmitted by tsetse fly. South America, Chagas disease transmitted by the redovid bug. Toxoplasma gondii, ingestion of oocysts from cat feces. Remember your torch infections from term 2? One of them is toxoplasma, right? So you can have neonatal toxoplasmosis. So what's the high yield point here for the extraintestinal blood and tissue protozoa? First, you need to know the vector. How is it going to be transmitted, right? Just like the intestinal one. Well, the intestinal ones are transmitted by oocysts, right? Or trophozoites directly. For this one, you need to know the vector. And you need to know the diseases that it causes, right? Let's summarize this slide. Plasmodium, 4 species that infect us. Falciparum, the most deadly, right? It can cause hemorrhage, hemorrhagic fever. Vivax and ovale, they have hypnozoites. And you have Plasmodium malariae, tertian fever. Leishmania, transmitted by what? Sandflies. It can lead to what? Visceral and cutaneous leishmaniasis. Trypanosoma. In Africa, you have the African sleeping sickness. It is transmitted by what? Tsetse fly. In South America, you have Chagas disease. It is transmitted by what? The redovid bug. Toxoplasma gondii, transmitted by oocysts from cat feces. So tell your pregnant woman not to take out the kitty litter box, okay? Because you might get oocysts from the cat feces there. It can lead to what? Cause of neonatal toxoplasmosis. This causes ring-enhancing lesions on the brain. If you look at the scan of the brain. Okay, put a star here. Plasmodium, the generalized life cycle. I want you to focus on 3, okay? So when the mosquito injects you with this, it's in the form of sporozoites. Okay, that's high yield sporozoites. So the infective stage is sporozoites. It goes to your liver, right? It goes to your liver. If it's Vivax or ovale, it will going to become hypnozoites. It's just going to stay there. It will going to sleep, right? Hypnozoites there, replicating, replicating, replicating. If it's not Vivax or ovale, if it's falciparum or malariae, it becomes the schizons and converted into merozoites, right? So the infective form is sporozoites. In the liver, they become merozoites. And this is the one that will gonna infect your RBCs, your red blood cells. Once in the RBCs, it becomes a schizont again and gametozoites. Okay, so what's high yield point here? Plasmodium via mosquito bite, female Anopheles mosquito, infective form, sporozoites. In the liver, it becomes what? Merozoites. This is also the infective form for your red blood cells, the merozoites. And after replication in the RBC, it becomes what? Gametozoites. Okay, so sporozoites, merozoites, and gametozoites. I don't know if that's a correct pronunciation, but know those 3. Okay, hepatic in the liver, it's a merozoite that infects the RBCs. After the infection of the RBCs, it becomes gametozoites. Leishmania and trypanosoma. Put a star here. This is high yield. Leishmania via sandfly, promastigote. In the mammal, it's a mastigote. Know the terms, basically. For the American trypanosome and the African trypanosome, different vector. For the American trypanosome, we have the redovid bug, right? For the African trypanosome, we have the tsetse fly. Okay, tsetse fly. Same infective form. epimastigotes for both the American trypanosomes and African trypanosomes. Epimastigote. They become tripomastigote in the mammals. Okay? Let's summarize this slide. Leishmania, what's the vector? Sandfly. Promastigote. It infects a mammal. The promastigote becomes what? A mastigote. American trypanosomes. What's the vector? Redovid bug. Infective form, epimastigote. In the mammal, it becomes what? Tripomastigote. African trypanosomes. Your sleeping sickness. What's the vector? Tsetse fly. What's the infective form? Epimastigote. In the mammal, it becomes what? The epimastigote becomes tripomastigote. So those are your high yield. This slide. So let's look at this one. So the clinical manifestations, as I've said, you have cutaneous and visceral. Leishmaniasis, look at this, the cutaneous form.
Question about the previous, like, infectious form.
So when they bit you, right?
Yeah, so both of the infectious form in Chagas and the African sleeping sickness are epimastigote.
Yes, epimastigote.
And then the intermediate host would be the tripomastigote.
Yes, the mammal, yep. Okay, you're welcome. That's high yield, okay? Okay, for Chagas disease, look at the image. Romagna sign. You're going to learn this one, I think, term 4 or 5. So you have a swelling of the eyelid in your acute Chagas disease. Next term, you're going to learn about the different manifestations. But for now, if they're going to give you a picture of a child with a swelling on the eyelid, think about Chagas disease. If it's, look at the picture on the left side, right? So we have the visceral leishmaniasis, right? And then the cutaneous leishmaniasis. Don't forget, this is, what's the vector for leishmania? Sandfly. Now, Naegleria fowleri and Acanthamoeba. If you're like me, you love swimming. You want to go to the lake, Lake Champlain, for example. It's a freshwater lake, and you're swimming. Then you've got this terrible headache, right? And then after a couple of days, you die. Why? One of the primary causes of primary acute meningoencephalitis is your Naegleria fowleri. That's high yield, fresh water. Okay? How does it infect? Let's go back to term 2. How does it infect a human being? Where does it go through?
The cribriform plate.
Exactly.
The brain.
Exactly. So cribriform plate and then through your brain. So Naegleria fowleri, where can we find this one? Freshwater, such as lakes and streams, right? How does it invade the brain? Via the cribriform plate. And then what's the disease? Primary acute meningoencephalitis. You have inflammation of both of your brain and both of your meninges. Next, another one is Acanthamoeba. Acanthamoeba species. It is found also in freshwater, such as your lakes and streams. High yield. It contaminates your what? If you're using something for your contact lenses, the liquid, right? For your contact lenses, it contaminates that one. So that's high yield. It contaminates fluid used to treat contact lenses. So what? You said it can cause keratitis, resulting in eye pain, redness, tearing, and loss of vision. Do I look at these pretty pictures? Do I need to know them? Yes, they might show you images, right? So this is a trypanosoma rosei. This is trypanosoma cruzi. You can see the tripomastigote in mammals now. Leishmania here, you can see the amastigotes, right, in mammals. And then this is a trophozoite for your Plasmodium vivax in a blood smear. I think next term we're gonna go deeper into these diseases. But for right now, please recognize the images because we're gonna give you images. All right, who watched this movie? Did you watch Parasite by Bong Joon-ho?
Yeah.
So watch this movie. It's one of the greatest movies of all time, I think. So we're gonna talk about parasites. Okay. Do I need to know the names? Yes, of course, you need to know them. The difference between a roundworm, a nematode, and the flatworms. So nematodes are roundworms. Trematodes are your flukes. Cestodes are your tapeworms. Okay, what's the physiology? They're parasitic, right? They're multicellular. How do they acquire nutrition? Ingestion or passive absorption. They reproduce sexually and asexually, and they produce large number of eggs per day. Know the type and most common route of infection and the infective forms. For nematodes, it's all eggs. Trematodes, Cercariae. Cestos, you have proglutids. It's not important. Let's go to different kinds. Okay, put a star here. Nematodes, the general features of your round worms. They have cylindrical bodies. They have a complete digestive system. You have separate male and female sexes. They're not hermaphrodites. They have both male and female species, right? And the eggs, they have a characteristic shape. They have a cap and a cell wall on both ends. You call this one operculum. Highlight that one. So for eggs, they have a characteristic shape, cell wall, density, and cap. They have an operculum on one or both ends. Nematodes, let's summarize. Nematodes, complete cylindrical bodies, complete digestive tract. They are separate male and female sexes, which mate together and produce the eggs. And the eggs has a characteristic shape, cell wall, and a cap. We call the cap operculum on one or both ends. Life cycle, basically, you ingest them as an egg in the body, you become adults, basically. Okay, let's go to the different kinds of your nematodes. Ascaris lumbricoides, your roundworms. Go to YouTube and type Ascaris lumbricoides. You're gonna find a lot of surgeries due to this roundworm. They actually block your GI tract, and it looks like spaghetti, to be honest with you, like the pasta with the spaghetti. You watch it on YouTube, okay? So Ascaris, your roundworm. It is found worldwide. What's the mode of transmission? Fecal-oral, right? It causes what? Since it's fecal-oral, it's a roundworm, it causes an increase in your eosinophils. So eosinophilia. Enterobius vermicularis, your pinworm. It is found mainly in children. It produces anal itching. There's anal itching, then it's Enterobius vermicularis. What's the test that you're going to do? Scotch tape test. Why do you have pruritus? Why do you have anal pruritus in Enterobius? Based on the life cycle. Anyone? Why do you have anal pruritus in Enterobius vermicularis infection?
That's where they live, right?
You have anal pruritus, right? Because the way you ingest them as an oocyst, right? And then when they mature, they migrate to the anal region, actually. That's why you have an itching there, okay? So Enterobius. So let's summarize. Ascaris lumbricoides. Mode of transmission, fecal-oral. Like most of the intestinal nematodes, they are fecal-oral. It causes what? Eosinophilia, because it's a roundworm. Enterobius vermicularis, a pinworm, found mainly in children. It causes what? Anal pruritus. What's the test? For this, Scotch tape test. How are we going to treat most of these intestinal nematodes? Just use bendazoles, albendazoles. Your mebendazole, your albendazoles for this. Trichuris, whipworm. Look at the image again. Imaging is important. Imaging of the actual organism and the imaging of the egg. Okay, they might just give you an image of the oocyst. Okay, so for the trichuris, they call this one Trichuris trichuria, I think, if I'm not mistaken. It is prevalent in what? Warm, humid conditions. It causes diarrhea, rectal prolapse, and anemia. That's high yield. Ancylostoma and Necator americanus, your hookworms. They're the major cause of what? Anemia in the tropics. Look at the image, look at the eggs. Strongyloides infection. More severe in immunocompromised people. That's why you don't go, so that's why you don't walk barefoot when you go to the beach. Why? Because of strongyloides. Okay? That's just a public service announcement. So summarize. Intestinal nematodes. 1. Trichuria. What's the mode of transmission? Via soil. What does it cause? Diarrhea, rectal prolapse, anemia. Next, Ancylostoma and Necator, hookworms. How do they infect the human being? The Ancylostoma and Necator, hookworms. Anyone?
Tissue penetration.
Tissue penetration, right? Exactly. Major cause of anemia in tropics. How about strongyloides? How do they affect a human being? Same thing as the hookworms, right? Tissue penetration. More severe in what? Immunocompromised people. How do we treat them? The intestinal nematodes, bendazoles, albendazole, mebendazoles. Tissue nematodes. Look at the image again. Look at the eggs. Toxocara. You have Toxocara canis. This is a worldwide infection of dogs and cats, right? So you ingest an embryonated egg from a dog or a cat feces. It can cause what? Highlight this one. Visceral larva migrans for the Toxocara. Trichinella trichuris. The trichinella. Consumption of infected and unfrozen or poorly cooked meat. And then the encysted larva, where does it go? To your muscles. Look at this one. On the right side, lower right side of the slide, you can have an encysted larva in your muscles. It's very, very painful. So let's summarize. Toxocara. What's the mode of infection? Ingestion of embryonated eggs from dog or cat feces. In children, it can cause what? Visceral larva migrans. Trichinella. Trichinella, what's the mode of infection? Consumption of infected and unfrozen or poorly cooked meat. And then it insists where? In your muscle, striated muscle. So it can cause muscle pain. So there's another, put a star here. A generalized life cycle of your nematodes. Basically, undercooked food, fecal-oral route. And then the larvae, right, migrates to something, and then it penetrates and insists to your body. Okay, filarial nematodes. There are 3 that you need to know. Onchocerca volvulus. This is transmitted by the black fly. High yield. It can cause visual impairment. What's the disease that this filarial worm can cause? What do you call that disease? For Onchocerca volvulus. Did you talk about river blindness? No?
Not yet.
Okay, so always associate Onchocerca volvulus with river blindness. Okay, river blindness, Onchocerca volvulus. It can cause visual impairment, blindness, and severe itching of the skin in infected individuals.
Nick, can I ask a question about trichinella? Since is it considered officially an intestinal nematode since it is transmitted through food, but the larvae is found in the tissue, so it could also be considered a tissue nematode?
We classify it based on the mode of infection, the infectivity. So basically, it's an intestinal, right?
Okay.
But you said, but the process of encystment occurs in the muscles.
In the muscle.
Yes.
And it's found as a larva in the muscle.
Yes,
okay.
in the muscles. Yes.
And the
so when
worm
they give you a slide of the specimen and you see that it is muscle, it is striated, muscle, right?
Mm-hmm.
So
Gotcha.
they're
Thank you.
going to give you so you need to look for each of these, the one that we talk about, the protozoans and all of those good stuff. Look at the imaging. The images are there not to confuse you. We have this notion about why do they put image there? I mean, it doesn't help me. No, actually, it helps you. They're gonna put some signs and symptoms on the vignette, and you're thinking, this is this is a nematode or something. But what kind of nematode? You're confused now, right? So they're gonna give you an image. So the imaging helps. So please make time to look at the image, look at the adult form of that worm, and then also look at the eggs, because they might just give you the eggs, right? The oocyst form. So look for the adult form, pay attention on the adult form and the egg form, the oocyst form, okay? So the imaging is there to help you, all right? So please make sure you know the images. That's very important for this exam. Okay, so filarial nematodes, what time is it? 11. Okay, Onchocerca, rival blindness. Wucherera bancrofti, high yield, lymphatic filariasis. Look at the image on the lower left side, right? So you have this one, your Wucherera bancrofti. Look at the right side. See this inflamed lymph here? Oh my God, this kid can't walk, right? So you have lymphatic filariasis, Wucherera bancrofti. Another one that can cause this is Brugia malayi, but that's low yield. So when you think about elephantiasis or lymphatic filariasis, think about Wucherera bancrofti. Filarial nematodes, put a star here. So they live in as a human host, right? So first, they live in the fly. Mosquitoes, black flies, horse flies, or deer flies. And they develop to infective larvae in your insect host, right? Then what's the mode of transmission? Taking a blood meal, all right? And then in the human host, they live in the lymphatics, subcutaneous tissue, skin, and your cavities. And then the microfilariae, they're in the blood and subcutaneous tissue, and then ingested back by your biting insects. Okay,
So what form are they in the human?
they are microfilariae in
Okay.
your blood and your subcutaneous tissue.
Thank
Okay.
you.
Yep. So trematodes, flukes, put a star here. What are the features, right? One, bed and digestive system. What do I mean by bed and digestive system? This is yucky. The nutrients are taken in, digested, and then expelled through the same opening. Can you imagine that one, ladies and gentlemen? Thank God we don't have a dead end digestive system. Now they're classified as flat worms. High yield. They involve an intermediate. What is that intermediate in their life cycle? The mollusk and then a vertebrate. Okay, so let's summarize. Trematodes. Characteristics: dead end digestive system, meaning food goes in, food goes out, one way. There's only one. They're classified, both of them are classified in one phylum, your platyhelminths or your flatworms. Their life cycle involves more than one host. So you have a mollusk and a vertebrate. That's high yield. For this one, remember the snail, right? Snail, and then you eat the snail, and then everything happens, right? But for this one, remember, you have a snail. This is fasciola. Okay, that's not important. I think what's important is this. Put a star here. So different kinds, right? Fasciolopsis busci. Look at the image again. Imaging, imaging, imaging. Look at the image on the right side. The adult form and the egg form, right? So this is most common in what? Humans and pigs in Southeast Asia. One of the largest trematodes to infect man. High yield, it causes inflammation and hemorrhage of your small intestine. That's your intestinal fluke. Paragonimus westermani, look at the image again, right? Acquired by what? Ingestion of metacercariae in raw or pickled crustaceans. Lung fluke, why? Because it migrates into the lungs actually, your Paragonimus westermani. There are different kinds of species of Paragonimus, but Paragonimus westermani, it goes to your lungs, okay? I forgot to tell you, just remember that your ancylostoma and Necator, they can also migrate to your lungs, okay? It can also cause respiratory symptoms. So what's the clincher? Is it Paragonimus westermani or one of your hookworms? Imaging. They're gonna put some images there. I know I sound like an old person yelling things, right? But please know the imaging. It will gonna help you. Next, fasciola hepatica, like an old broken record. Fasciola hepatica, liver fluke. Liver fluke. So how are we going to acquire the liver fluke? You ingest them. What's the infective form? Metacercariae, right? And then they live in what? Intrahepatic bile ducts of the liver. That's also high yield. So if they live in the intrahepatic bile ducts of the liver, this can cause what? Let's go back to term 2. It can cause what? Anyone? Intrahepatic bile ducts of the liver. It can cause what?
Bile obstruction?
Bowel obstruction, yes. So you can have cholangiocarcinoma. Exactly. So it can cause bowel obstruction, right? Okay, next, Clonorchis sinensis, the Chinese liver fluke, acquired by ingestion of infective Metacercariae in raw or pickled fish. Okay, trematodes, flukes, let's summarize here. Fasciola hepatica. Fasciola, think about liver. They look like a liver here, right? This liver fluke on the right side, the image here, it looks like a liver, right? What's the mode of infection? You ingest via what? Metacercariae. That's high yield Metacercariae. And then the adult trematode, once inside the body, they become trematodes now. And they live where? Intrahepatic bile ducts of the liver. That's high yield. It can also cause severe anemia in humans, high yield. Next, if they're going to give you geographic origin, like for example, someone in China, Japan, Korea, think about Clonorchis sinensis, right? This is acquired through what? What's the infective form? Metacercariae. What's the route of infection? Fecal-oral. Ingestion of the Metacercariae in raw or pickled fish. So any sushi lovers out there, watch out for Clonorchis sinensis. Schistosoma species. Look at this one. It's a trematode. It exists not as a hermaphrodite. They have 2 species. You have a male and a female worm. Do I need to know the differences of each species? Yes. Look at the image at the bottom. They're going to give you a picture of the egg. Differentiate Mansoni, Hematobium, and Japonicum. What can you say about the 3? Look at the image. The Mansoni, Hematobium, and Japonicum. How are you going to differentiate them? Just by looking at them. Hello?
Round oval shape, like round versus oval shape in the presence of the spine.
Exactly. So the presence of the spine, that's the key. So in S. mansoni, you can see where is the spine in S. mansoni? The lateral, right? You have a lateral spine in your S. mansoni. How about the hematobium? Where is the spine? At the end, right? At the end, you can have the spine. What's high yield here? Look at the imaging, the eggs on the imaging. Check the spine to differentiate whether it's Mansoni, Hematobium, or Japonicum. And where does it localize? That's also high yield. For Japonicum and Mansoni, they localize in the mesenteric veins after entering the circulation. So, as I've said, much prone to liver enlargement. You can have enlarged liver due to blockage of your mesenteric veins. So you can have hepatomegaly for this one. Hematobium, where does it localize? Bladder. So it localizes to the bladder. So most common cause of transitional carcinoma, or urothelial carcinoma, its high yield is Schistosoma hematobium. Don't forget what they look like. Okay?
Japanosium not have a spine, or I just don't see it.
I think the Japonicum is just round. Yeah.
Okay.
Yep. And don't forget, where can we get these eggs? The Mansoni and Hematobium, it's on the stool. Contrast this one with your Japonicum and Mansoni, I'm sorry, can be found in the stool, and the Hematobium can be found in the urine, right? Because most common cause of transitional carcinoma or your bladder carcinoma is Schistosoma hematobium. Japonicum and Mansoni, most common cause of hepatomegaly. So it can cause hepatomegaly. Why? Enlargement of the liver, because it localizes in the mesenteric veins after entering the circulation. All right, generalized life cycle. Don't forget the snail. What's the infective form? Metacercariae. If you ingest them, right? That's the point here. Okay, cestodes. Put a star here. You need to know the general features. You have they have a flat segment. We call that one what? Proglottids. Highlight that one. That's high yield proglottids. And they have a head. What do you call that head? Highlighted scolex. That's high yield, the scolex. What's the importance of the scolex? It contains suckers or hooks used to attach to the intestinal epithelium. Remember, the worm has no digestive tract. So if they don't have a digestive tract, how do they absorb nutrients? They absorb nutrients directly into the proglottids and to your scolexes, right? Do they have a male and female species like your nematodes? No, they are hermaphroditic. So tapeworms are hermaphroditic. Each proglottids produce sperm and egg that are engaged in self-fertilization, right? And what's the definitive host for your tapeworms? Humans. Humans are the definitive host. Signs and symptoms, of course, you can have that one, but let's look at the specifics. So intestinal tapeworms, your Taenia solium or Taenia saginata. Taenia saginata, mode of infection, ingestion of contaminated or uncooked beef, so it's fecal-oral again. And then you have a common infection but causes minimal symptoms. Look at the image, look at the eggs. Taenia solium, pork tapeworm. You have ingestion of your contaminated or uncooked pork that contains cysticerci. So it causes what? High-yield cysticercosis. What is this? When the cysticerci insist in your muscles and in the brain, which can lead to epilepsy. So for Taenia solium, think about brain. So they go to the brain. Most common cause of epilepsy. All right, your Taenia solium. Infective stage, again, don't forget cysticercus larvae. What's the method of infection? You eat them. If you eat beef, it's saginata. If you eat pork, it's solium. And you have the rest in an adult. How are we going to diagnose them? Eggs. Or sometimes you have proglottids passed into your feces. That's high yield for this one. Put the star here, know the infective stage, cysticercus. Method of infection, eating the cysticercus. What's the diagnostic for Taenia? Eggs or proglottids in your feces. Echinococcus granulosus and Echinococcus multicularis. They're hydatid disease, right? Because when you look at the image, they form hydatid cysts. Look at the imaging again here. They may develop in the human host and cause space-occupying lesions in several organs like the liver and the brain. Hydatid cysts, think Echinococcus. That's high yield. Hydatid cysts, think Echinococcus. Fungi. They're eukaryotic. Put a star here on this table. The difference between fungi and bacteria, remember the fungi, they have cytoplasmic organelles, contrast with bacteria. Their membrane sterols are ergosterol, right? While bacteria doesn't have any membrane sterols, except for a mycoplasma, which has teichoic acids. And metabolism for fungi, they are mainly aerobes, and they are common in many pathogenic species. They exhibit what we call dimorphism. They can exist as a mold, or as the yeast. Fungal cell wall. The defining characteristics of the fungal cell wall is this. They have chitin and beta-1,3 and beta-1,6 glucans. Highlight that one. Beta-1,3 and beta-1,6 glucans, because next term we're going to learn about echinocandins, which targets the ergosterol. It targets this beta-1,3 and beta-1,6 glucans. So they have ergosterol instead of cholesterol in their membrane. That's also high yield. In terms of morphology, they can be unicellular or multicellular. As I've said, they can be a yeast or a mold. How do they decide which one is which? If I'm a fungi, how do I know if it's time to be a yeast or it's time to be a mold?
Temperature.
Exactly, temperature. Which one is which?
Yeast in the heat, mold in the cold.
Excellent. Yeast in the heat, mold in the cold. Someone is watching a sketchy. Very good. So hyphae, so the multicellular fungi, they connect together in long strands, right? They're known as your hyphae. If the hyphae becomes tangled, they're called mycelia. So know the definitions, please. Some of them have septate or aseptate. What do I mean by septate or aseptate? They have walls between neighboring cells. Don't forget that the nuclei of a fungus is haploid, right? They are haploid except when they generate. Okay, so this is it. Dimorphic fungi, yeast and mold. Reproduction. What's for fungi? How do they reproduce? They reproduce asexually by mitosis or sexually, and they form spores. Asexual reproduction is called budding. High yield, unequal cell division. Okay, so they use budding. There are different kinds of mycoses. Okay, let's go to the different types. Okay, fungal classification: Ascomycota, Basidiomycota, Zygomycota. No, that's not it. Okay, here it is. This is much more important. Fungal classification, or ascomycetes, right? They form sexual spores in what? In an enclosed sac. High yield is Candida albicans. Candida albicans. Most common cause of yeast infection in humans is Candida albicans. Pneumocystis jirovecii. This is most commonly caused of pneumonia in AIDS patients. Your Pneumocystis jirovecii, especially if their CD4 is less than 50. For Basidiomycota, think Cryptococcus neoformans. Cryptococcus neoformans. Most common cause of brain infection in HIV AIDS patient. Okay, in CD4 count less than 50. Cryptococcus neoformans. Zygomycota, they have sexual spores in a sac called zygosporangium. Okay, so let's summarize. Ascomycota, important species, Candida albicans and PJP. Basidiomycota, they have a basidium. An important species is Cryptococcus neoformans. Zygomycota, zygomycetes. Most important pathogen, Mucor species and Rhizopus species, right? Okay, for the Mucor and Rhizopus, think most common cause of fungal infection in diabetic patients, especially type 1. They have diabetic ketoacidosis. Okay, DKA. When in the vignette, it says DKA, right? And they're having fungal infection. The most common cause is Mucor, the Zygomycotas, your Mucor and your Rhizopus. Superficial mycoses. So you have Pterisis versicolor, Tinea versicolor, caused by what? Malassezia furfur. So how does it look like on the skin? Your Malassezia furfur. How does it look like on the skin if you have an infection of these? What are we thinking?
Is it usually hypopigmented?
Actually, both hyperpigmented and hypopigmented for the skin. What's the prep that we're going to use for most of your mycoses? KOH, right? That's your KOH strain. So when you look at the Malassezia furfur, right? What's the characteristic? What do you call this one? Characteristic image that you can see from Malassezia furfur on the KOH prep stain. If you watch Sketchy, you know this one. If you watch Sketchy, you should know this one. Malassezia furfur, spaghetti and meatballs appearance. So guys, for now, please make sure you go. I think Sketchy is very, very helpful. I'm a visual learner, right? If you're a visual learner, please make sure you watch Sketchy Micro. Train your brain now to watch those videos. You have Sketchy Micro, and for next term, you have Sketchy Path and Sketchy Farm. So right now, watch Sketchy Micro to train your brain how to process the images there. Why? Because when you go to term 4 and term 5, it's much more helpful for you, I think, if you're a visual learner, right? That's the caveat. Visual learner, do Sketchy, please. It will gonna help you in the long run. So for tinea nigra, your horte, Wernicke, it causes skin to darken. So look at the image. It causes the skin to darken, right? Black Pedra, superficial infection of the hair. What's the causative organism? Pedra horte, right? Then you also have onychomycosis. When you see onychomycosis, think fingernails. Onychomycosis, fingernails. So you have your trichophyton rubrum, and trichophyton mentagrophytis. What's the treatment for this? It also starts with T, terbinafine. So dermatophytes, they don't penetrate deeper, right? They're just on the epidermis of the skin. So let's summarize here. Superficial mycosis. What's the high yield point? 1. Tinea versicolor. What's the causative agent? Malassezia furfur. How does it look like on the slide? KOH stain, spaghetti, and meatballs. Tinea nigra. Caused by what? Horte Wernicke. What can be seen in the patient? It causes the skin to darken. Black Pedra and white Pedra. Causative organism? Pedra horte. What does it do in a human? Superficial infection of the hair shaft. Onychomycosis? Think about fingernails, infection of the fingernails. Examples of organisms? Trichophyton rubrum, Trichophyton mentagrophytes. And the dermatophytes, they're only on the superficial layer of your skin. They do not penetrate deeper, okay? Oh, what's going on? Cutaneous mycosis, so your tinea. So look at the image on the right. Do I need to know the differences? Yes. So barbae, think about beard. So these are your skin infections, right? Cutaneous mycosis. Tinea barbae, beard. Pedis, foot. Cruris, groin. These are Latin terms. Capitis, scalp. Corporis, trunk. Right? So please make sure you know the differences. How do they spread? The superficial mycosis or cutaneous mycosis, they spread via scratching. They're very, very itchy, right? So they don't spread systematically. Remember, they are just cutaneous. These are your dermatophytes, right? However, what's the danger for the cutaneous mycosis? So you're scratching them, so you can develop secondary bacterial infections, high yield. That's a danger. You can develop a secondary bacterial infection. Subcutaneous, now we're no longer at the layer of the skin, right? What are the layers of the skin, by the way? Let's go back to term 1. Remember Californians like girls in string bikinis?
Corneum, lucidum, granulosum, not granulosum, come let's get sunburned, spinotum, and basali.
Exactly. Excellent. Excellent. So we're now in the subcutaneous layers, which involves the deep viscera, right? What's high yield? Sporotrichosis, causative organism, Sporothrix schenki. So this one, it follows the lymphatics. What population can cause this? The Sporothrix? Rose gardener, right? So if this is called rose gardener's disease. So when you're pricked by a thorn, careful, it might have Sporothrix schenki. And look at the image here on the right side. So it follows the lymphatic system. Okay, look, it's ascending infection. It ascends the lymphatics. Next, mycetoma. It's a granulomatous inflammation, and it has pigmented nodules that may drain through sinuses and produce colored grains. But the worst of them all is the chromoblastomycosis. You have warty, pigmented lesions. Let's summarize. High yield points. Sporotrichosis, causative agent, Sporothrix schenki, rose gardener's disease. This infection, it follows the lymphatics. So it ascends their lymphatics. Next, mycetoma. It's a granulomatous infection that extends beneath the subcutaneous region of the bone. It may drain through sinuses and produce colored grains. The worst of them all, the worst of the subcutaneous mycosis, chromoblastomycosis. You have warty, pigmented lesions. Opportunistic mycosis only for weakened immune system. Candidiasis, Candida albicans. High yield, white pasty colony with a yeasty odor. Most common cause of what? Thrush, vulvovaginal yeast infection, and cutaneous candidiasis. Aspergillosis, a common airborne soil fungus, most common in immunocompromised individuals. Next term, we're going to learn about ABPA. Cryptococcus neoformans, most commonly found where? Pigeon roost. Usually infects the lungs, most common infection in immunocompromised patients. All right. Zygomycosis, as I've said, your rhizopus in mucor, most commonly cause of fungal infection in patients with DKA, diabetes. And then your PJP, your pneumocystis jirovecii, it causes pneumocystis pneumonia, especially in AIDS patients. Which one is pseudohyphae? When they give you a vignette, it says there's a pseudohyphae.
Candida albicans.
Candida albicans. Most common cause of pneumonia in AIDS patient whose CD count is less than 50. Is it candidiasis?
Pneumocystis jirovecii.
Exactly. Pneumocystis jirovecii. Most common cause of brain infection in someone who has AIDS, HIV/AIDS. Now we're looking at Cryptococcus neoformans. So my advice to you here is watch Sketchy because they give you the different characteristics of each, right? How are we going to diagnose fungal infections? You can read those things. KOH strain, right? What's the medium for culturing fungi? What's the medium? Sabarodes, dextrose, agar. There are different kinds, right? There are different kinds of agar for each of them. Pseudohyphae, as I've said, Candida. Low angle branching, Aspergillus. Right angle branching, Zygomycetes. That's high yield. Please know that one. Low angle, Aspergillus. Right angle branching, Zygomycetes. Right? So spores. What's high yield here? Arthrospores and blastospores. What's the difference between arthrospores and blastospores? Arthrospores, they become conidia as they break off from the end of your hyphae. Contrast this one with blastospores, which are produced by budding. Okay? So arthrospores, they become conidia as they break off from the end of a hyphae. Blastospores, they are produced by budding, such as in the ends of a phyllides or simply from the sides of your hyphae. And chlamydospores, they are thick-walled spores capable of surviving adverse conditions. Do we need to know the difference between macroconidia and microconidia? Well, it just depends on the size of your spores, right? Fungal cultures, I've said, saboros, agar, because fungi are dimorphic. They can be oval, spherical, budding, or encapsulated. Saboros, agar, yada, yada, yada. 10%, what's the stain that we're going to use? 10% KOH stain. What's the agar that we're going to use? Saboros agar. Okay, that's different. When are we going to use the silver stain? High yield, PJP, your Pneumocystis jirovecii, right? So you do a bronchoalveolar lavage of a patient, and then you stain this. If you suspect pneumocystis, you stain this one with your silver staining, right? What's the stain used for Cryptococcus?
Like Gymsa?
For Cryptococcus. What's the stain used for Cryptococcus neoformans? So that you can see the capsule. You can see beautifully the capsule. Anyone?
Would it be a gram stain?
Nope.
The India ink?
India ink, excellent. India ink for your Cryptococcus, so that you can see well the capsule. Remember, it's an encapsulated organism, so that you can see the capsule really, really well. Testing fungal serology, you can do LISA or ELISA. Can you immunize? Can you give vaccinations for fungal infections?
I'm sorry, what was that stain again?
India ink for Cryptococcus neoformans. So can you give vaccines for someone who has fungal infections? Yes or no? Can you? No, of course, right? It's don't give antibiotics in someone who has a viral infection. It doesn't work. Okay. All right. Let's have a break for a minute. Okay, like 5-minute break. Then we go over with immunology. Is that okay with everyone? You just need to drink some water.
Yes.
Yes.
Okay.
All right. Let me stop sharing here. Stop sharing.